Hello, welcome back to another WIMBA presentation. This is Professor Young, and we are dealing with part 2 of 2 for the digestive system. What is before you, or I should say what we covered during the first session here, was the anatomy and some of the physiology involved of the digestive system up to the stomach. What we now need to consider is everything else that comes after the stomach. We're going to start our discussion here with the accessory structures, such as the gallbladder and the liver and the pancreas, and move into back into the main structures of the digestive system, and that being the intestines and so on and so forth, and what really happens during the intestines as far as absorption and reabsorption, and then finally into the large intestines and the colon, aka the colon. What I should mention here before we go any further, at the end of the last session, once the stomach is done with its churning and it's done with peristalsis and it's released its hydrochloric acid and it's released gastric lipase and it's released pepsinogen into pepsin and it's working with lingual lipase and everything else, you have this lumpy pea soup type of leftover material of digested goodness, and that is going to be squirted, if you would, from the stomach in a very controlled fashion into the duodenum of the small intestines. And the way that happens is through a structure known as the pyloric sphincter. And that structure that we are squirting through, and we'll probably come back to this again when we talk about small intestines, is called chyme, C-H-Y-M, chyme. And that's basically all the digested material up until that point. Remember, nothing's really been absorbed here or reabsorbed back into the system at all. That's going to happen during the time it spends within the small intestines. All right, so with that out of the way and with that said, let's get back to the accessory structures here. We're going to start with our look at the liver as well as the gallbladder. Both of these are going to play a role into one another as they lead into the pancreas. The liver's whole purpose here is to secrete bile, and that bile is actually going to be incorporated into the gallbladder and will basically aid in lipid absorption into the intestinal tract, and we'll talk a little bit more about that. When we look at the flow of bile coming out of the liver, it starts with the bile canaliculi, or I should say, yes, that is true. The bile canaliculi found within the liver, these small canals that are going to contain this bile material. From there, it's going to travel into the bile ductules, into the right and left hepatic ducts, down into the common hepatic duct, the cystic duct. That cystic duct is where the gallbladder meets in with the bile being released from the liver and kind of gets sucked back up into the gallbladder. And we'll talk about why that is in a couple minutes here. But then from there, it goes down into the bile duct and joins forces with the pancreas in what we call the hepatopancreatic ampulla. And basically, this is a region within the pancreas where we have the secretions of bile coming out of both the gallbladder and the liver, meeting with pancreatic juice, the combination of pancreatic enzymes being released from within the pancreas, and as it leads into the small intestines. It's going to do that again through a very controlled mechanism called the sphincter of Oddi. And the sphincter of Oddi is there to, again, allow for the passage of both pancreatic juice as well as the bile coming from the liver and the gallbladder into the small intestines. It's got to be regulated, and it's got to be timed with the release of chyme, because as it empties into the small intestines, specifically the duodenum, that first part, the first couple of centimeters or inches of the small intestines, it needs to neutralize. Here's the key: it needs to neutralize hydrochloric acid. Because if we were to leave the pH as low as it is within the stomach, we would have major issues coming into the small intestines. We would have irritation. We would have redness. We would have inflammation. We would have eating away of the lining within the small intestines. And so what all this stuff does coming through the sphincter of Oddi is neutralize. It buffers the chyme. It takes the sting out of it. It neutralizes or buffers the hydrochloric acid. So again, and we're going to look at a diagram of this. Don't think I'm going to leave you hanging like that, because I won't. But again, conceptually, mentally, and verbally, within the liver, we've got these small canals, these small channels called bile canaliculi. They're going to carry the bile into the bile ductules, down into the right and left hepatic ducts. They are going to join forces with the common hepatic duct, which is going to lead into the cystic duct, shoots the bile back upwards into the gallbladder. Stuff is going to be added within there, down into the bile duct, enters into the pancreas and joins forces with the pancreatic duct in what we call the hepatopancreatic ampulla. From there, pancreatic juice, bile, is secreted regularly and controlled through the sphincter of Oddi. All right, and that's going to regulate the sphincter of Oddi. Oddi regulates the release of pancreatic juice and bile into the duodenum, into that first region of the small intestines. Let's talk about the gallbladder here for just a few minutes. Bile, by definition, is made up of minerals. It's made up of cholesterol. It's made up of fats. It's made up of phospholipids. It's made up of bilirubin. Bilirubin is bile pigment that's going to be composed of or made up of decomposed hemoglobin. Hemoglobin that's worn out and it's gone off to hemoglobin heaven. And that's going to be added in, that's what we refer to as the bilirubin. It gives it that characteristic bile color, if you would, or bile pigment. And then we have the bile salts. Bile salts, another name for this, would be basically steroids that have been synthesized from cholesterol. And bile salts, technically, are the form of fats, lipids. Which is what steroids are. Steroids belong within the lipid category. And you should have remember that, or if you don't, go back to your A&P1 discussion of biochemistry and review that whole process there, because that's key and essential here. And again, the job of bile is really to aid in fat emulsification. Remember we said lipid breakdown happens or begins, is initiated, very small percentage of it, but is initiated within the stomach. Lingual lipase, gastric lipase, these two enzymes that begin to break down, or I think I did use the term emulsify, the fat droplets in the stomach. Well, what's going to further break down and emulsify that lipid or that fat is going to be the bile. And it's going to happen within the small intestines. And so that bile is initially produced within the liver. It's going to go into the gallbladder. And again, you're going to see where that's going to back up. You're going to see where that cystic duct is located and kind of forces that bile to go back up into the gallbladder. What we're basically doing is within the gallbladder is we're helping to remove some of the excess water, and we're removing some of the electrolytes. Again, when you're dealing with systems that are secretory in nature, or I should say excretory in nature, in other words, the end product is you're removing something from the body, you want to conserve as much as you can. The gallbladder is going to aid in that. It's going to absorb some of that basic water. It's going to absorb some of those electrolytes that are so important basis for maintaining homeostasis within the body. And so it concentrates that bile. It increases the effectiveness, basically, of it. Removes the water, removes the electrolytes, voila. From there, that bile will then leave the gallbladder, back down through the cystic duct, into the bile duct, and continue on its way. Urobilinogen is basically the further breakdown of bilirubin, but not until we get to the colon, not until we get to the large intestines. And it's bacteria regulated, and it's what gives feces that nasty brown odoriferous characteristic that we come to know feces by. I don't know any other way of saying that or trying to make that sound pleasant, so that's my shot at it. And so that's the job of the gallbladder. That's the job of the gallbladder. I did mention that we would go ahead and we would take a look at this from a picture perspective, and it is now picture time. You can see, and if you've got the slides for this, you can probably see it a little bit better, and I apologize for that. But you can see right here the hepatic ducts. That is what's labeled right there, and we are basically looking at these two structures coming out right there that are branching in. They are the hepatic ducts, both the right and the left, and they are going to join their forces to come into the common hepatic duct. Yes. The right and left hepatic ducts are going to come into the common hepatic duct. From there, following this along, you can see that we have the cystic duct that is going to lead right in there, and I'm going to kind of do away with that just so you can, but I wanted to point out where we're going to be looking at here. The cystic duct right there is going to lead, again, that bile back into the gallbladder, remove electrolytes, remove water, concentrate the bile. You can see, if you look closely, that it has chambers that's going to prevent the backflow of that bile. And then once it is secreted and concentrated, it will leave that. It will go down into the bile duct, which is right there. And that bile duct is going to lead down into the pancreas. And it's going to lead into that hepatopancreatic ampulla that you're looking at right there, more specifically. The ampulla is right there within that blue circle. That is where the ampulla is. And the purpose of that is to join the bile being released from the liver and the pancreas, the liver and the gallbladder, as it is flowing down into the ampulla. It is mixing with pancreatic juice from within the pancreas, and it is all being secreted into the duodenum through the sphincter of Oddi, which is where that last arrow just popped up. And so this area right in here is the duodenum. And actually the duodenum also then continues over into this area, and then eventually becomes the second portion and the longest portion of the small intestines, which is the jejunum. And so that's where we are structurally here. I'm going to do away with a couple of these arrows. The other thing I want to point out to you is if we clear or if we erase this slide, you will notice, and I want to draw your attention to right there. You will see that the pancreatic ducts come down and have a choice right here at this area. It will either continue to join down with the bile duct and go into the ampulla, or, and this is the key area here, or it can branch off here to what we call the accessory pancreatic duct. And that accessory pancreatic duct basically empties right into the duodenum. It empties right into the duodenum. Now, what would be the benefit of releasing pancreatic juice directly into the duodenum while bypassing the ampulla? Think about that. What is the liver secreting? If you are screaming at your computer right now, bile, you are correct. What is the purpose of bile? If you are screaming at your computer screen, fat emulsification, you are correct. So we are assuming that each and every meal we have enough fat present within the diet of what you are consuming that we need to have bile secreted. What if you have a very lean, healthy, vegetarian type of meal? You are chowing down and grazing on that salad. And you have one of those nice little light dressings, and it's low fat, and you don't have a whole lot of anything going on. And so therefore you don't need a whole lot of bile or any bile at all. Does it make sense for the pancreatic juice to necessarily pass through that hepatopancreatic ampulla? Or would it be more wise to have pancreatic juice, which is going to have its own digestive property, as well as helping in aiding in reabsorption or absorption? Would it make more sense to have that pancreatic juice have a bypass mechanism to where we don't need to release bile necessarily, but yet allow that pancreatic juice to go in and flow into the duodenum to again be able to help to neutralize hydrochloric acid and aid in further digestion? That would be the smart thing, and that would be the first choice. And that's exactly why we have that. The accessory pancreatic duct is there just for that reason, to bypass the ampulla, so that way we have a mechanism for buffering hydrochloric acid, for neutralizing the acidity coming out of the chyme being released through the pyloric sphincter. It's a great system. We don't always need high amounts of bile or any bile for digestion, depending on how much fat and how much lipids is present within the diet. And so if we don't need it, why secrete it? If you don't need it, why secrete it? That rhymes, and didn't even realize it was going to rhyme. And so if we don't need it, we're not going to secrete it. We're going to bypass it, and we're going to go through the accessory pancreatic duct. And that way you still allow for the function of the pancreas to neutralize the stomach acid, neutralize the chyme, and then allow the pancreatic enzymes, pancreatic lipase, and all the other good enzymes that are coming out of there to be able to do its job and do what it's supposed to do to aid in digestion. We talked about a little bit about with the intestines already, and you've looked at this in lab. We know that the pyloric sphincter is going to regulate the passage of chyme coming out of the stomach into the small intestines. Again, the first region that we are going to come into is the duodenum. That's going to bug me. From the duodenum, we hit this little flexion called the duodenal jejunal flexure. And really on the other side of this now, we have the jejunum. The jejunum is going to wrap and twist and curve and go all the way around until it reaches down into the ileum, which is the last final little bit of stretch of the small intestines, right there. And it leads into the ileocecal, what they're referring to here as a junction. Some books and some folks you might hear it referred to as the ileocecal sphincter. Either way, you're going to have a regulation or a regulatory effect of the chyme coming through the intestines that have now gone through all of this absorption, which we'll talk about in a couple seconds. But it's going to lead into the cecum, which is the first region, if you would, of the intestines. Notice that little old buddy that has no known function as of right now for humans, the appendix. That will then lead into, as we follow upward here, the ascending colon, which will then further lead into, and you're not seeing it here, but it will lead into the the transverse colon, down into the descending colon, around having a brain fart. Hold on one second. God, I hate when I do this. Geez, that's right. The sigmoid colon, could not think of it. Down into the sigmoid colon, and then down into the anus. And we'll look at a picture of that in a little bit. But as far as the small intestines go, it's going to join in with the into the cecum through the ileocecal sphincter. It's going to regulate the passage of the chyme going through the small intestines. Keep in mind, the longer the chyme is in the small intestines, the greater of a chance of complete absorption that we have. The faster it moves through, the less is absorbed, including the less water that's absorbed, and that's going to affect the consistency of the feces. That's all I'm going to say about that. I'm not going to go too much in depth with that one. But just be aware of that. Let's back up a little bit and go back into our intestines and look a little bit about what is the really making up the intestinal system and the intestinal tract. When we look at this, we're going to see some circular folds, we're going to see some villi, and we're going to see some microvilli. You should have been able to see this histologically. The circular folds are going to be located deep to the villi. Again, the villi are going to be lining all along right there. Underneath of the villi, you're going to have a layer of submucosal and mucosa. The submucosa and the mucosa is there to help to produce mucus, which is secreted through the villi to be able to, again, both lubricate the intestines and aid in the prevention of any kind of bacterial or viral infection getting into the tissue. Remember, we're still dealing technically with being outside of the body here. All right, this food stuff has never really entered into the body yet. We are now entering into this concept of being able to absorb some of these nutrients. but the food is not entering the body yet. That's a key distinction. And so you've got these circular folds. This is where a lot of your peristaltic contractions are going to be happening. Its job is to kind of pace out and regulate how fast that food is moving through, the chyme is moving through. The reason why we want to have these slow circular spiral patterns is because otherwise some food, let's say, may get stuck in the middle, which means it'll never reach the ends or the outer borders, if you would, of the intestinal tract. If it doesn't ever reach the lining of the intestines, it's not going to reach the villi. If it doesn't reach the villi and or the microvilli, this brush border, it's never going to have a chance of being absorbed. And so the job of those circular folds is to cause these circular patterns or spiral shotgun barrel type of patterns of muscular contraction that's going to swirl the chyme, so that all chyme at one point in time, will come into contact with the intestinal lining. All chyme, one point in time. Working on a wrap here or something like that. The absorptive cells, aka the microvilli, they're going to be lining the villi, and the goblet cells are there to secrete mucus. And so that's what's happening here. Let's take a little bit of an up-close and personal look with the villi, so you can kind of get an idea of what we are dealing with. What you are looking at here is a villi. If you look closely, if you look at these cells right in there, those cells are all lined with a brush border that we call microvilli. Now, in between those cells, such as right there, we have a goblet cell. Goblet cell is going to secrete mucus. And again, this has immune properties to it. Right? So it does serve a purpose. Those microvilli, that brush border, is there to help to increase the surface area. This is all about maximum surface area. The villi do a great job of increasing the surface area. These microvilli, though, exponentially increases the surface area. And so now this kind of even more surface to come in contact with, which means we can more efficiently and effectively absorb the nutrients that we need to have absorbed. Otherwise, by reducing surface area, by not having the brush border, by not having the intense amount of microvilli that we have, you limit the surface area, which means one of two things. You now have to eat more to get the same nutritional value, or you have to have a much longer digestive tract. You have to have a much longer, specifically here, small intestines. It's just not beneficial for our body plan to have anything much longer. We already have 20, 21 feet of small intestines in ourselves right now sitting there in your abdominal cavity. You've got all of that going on. And so you don't want to run into that problem. And so in order to shorten and shrink up the digestive tract, what we do is we exponentially increase all of the surface area, and it does that ever so well. We also see coming through here a series of capillaries, both arterial capillaries as well as venual or venous capillaries. As we absorb the nutrients, it's going to go right on into it. It's going to get absorbed into the arterial end. It's going to go right into the venules and be transported whatever it is that it needs to be transported. But then you see this green structure in there. And it's this lymphatic vessel. The lymphatic capillary, also referred to once that lymphatic capillary or lymphatic vessel enters up into the microvilli, we then refer to that as a lacteal. And that's what you're seeing right there. That's the lacteal vessel. Again, it's just a lymphatic vessel that has reached up into the villi itself. The lacteal is there to help to absorb lipids. This is where lipid absorption is really going to take hold and take place. So that way we can go ahead and distribute it to wherever it is that we need it using the lymphatic system. You also are going to see these intestinal crypts, which are, here's the entrance to the intestinal crypt. Here is the intestinal crypt, basically that space right there, and it is lined with these Paneth cells. The intestinal crypts secrete sucrase, they secrete maltase, which is going to, and it's going to secrete enteropeptidase. Right? So again, it's going to secrete sucrase, it's going to secrete maltase, aka enzymes that are going to help to break down carbohydrates, simple carbohydrates, simple sugars, and it's also going to secrete enteropeptidase. Enteropeptidase is going to be an enzyme that is going to help to activate pancreatic enzymes within the duodenum. And so that does serve a key structure and purpose, or it does serve a key purpose within the small intestines. Again, sucrase and maltase, they're going to be there to help to further break down simple carbs. And again, it's going to do that by activating, all three of these are going to activate pancreatic enzymes within the duodenum. Paneth cells, you're going to see that right down at the bottom. They're going to secrete lysozymes. Again, lysozymes, we've seen this within the oral cavity, being secreted by the salivary glands. But the Paneth cells are going to secrete these lysozymes that are again going to aid in bacterial resistance by aiding in viral resistance. And so the lysozymes help to do that as well. And then again, blood capillaries are going to aid in absorption. When we talk about breaking down carbohydrates, I already mentioned the fact that we've got sucrase and maltase that are going to aid in initiating or activating pancreatic enzymes, specifically pancreatic amylase. They're going to help to absorb glucose by using the sodium glucose transport proteins, the SGLT. We've talked about the sodium glucose transport proteins a little bit more in depth when we talked about the urinary system, but it's going to help to promote solvent drag. And so that way you have that extracellular fluid that is going to be become very concentrated with sugar. Proteins, we see a lot of these same terms coming over and over again, and that's a key concept to keep in mind. But proteins are going to be digested by protease, as we've already talked about, as pancreatic juice enters through the accessory pancreatic duct into the duodenum, it deactivates or buffers hydrochloric acid. Well, pepsin, in a sense, needed that acidic environment in order to work. The end result is inactivated pepsin. And so that's exactly what we see happen. Pepsin becomes inactivated in the duodenum, and then we see the next enzyme, trypsin, that's going to help to digest proteins, also within the small intestines. So we see both trypsin and protease working together here. The other thing I do, no, not yet. Next slide. The other thing I do want to mention here is a little bit about lipid digestion. Lipase is going to be specifically pancreatic lipase. Notice each region of the body, each region of the digestive system kind of has its own version of lipase. We started with lingual lipase, which was secreted in the mouth. We also had gastric lipase, which was secreted in the stomach. Now we have pancreatic lipase, which is going to be secreted from within the pancreas into the small intestines. And what it does is it basically helps to absorb the fat. Now, think about this. The fat here is entering into the duodenum, into the small intestines, as these large globs or globules of fat. Lingual lipase and gastric lipase is only going to do so much. It's going to help to emulsify it a little bit, but they're still too large to really enter in all by itself. This is where the bile comes into play. Remember we said that bile is going to aid in fat emulsification. It's going to aid in breaking down that fat. And so that's exactly what we see. The bile is added into the duodenum through the hepatopancreatic ampulla, through the sphincter of Oddi, and the bile basically surrounds itself around these large fat globules and create what we call miscels or micelles right there. And what ends up is that these are basically made up of the bile salts. The hydrophilic ends are outward. The hydrophobic ends are facing towards the fat globular itself. And it's basically carried to the absorptive cells. The microvilli brush border diffuses across the membrane. How is it able to get in there? How is it able to get in there? Think about this. What have I just said? What is this micelle? It's the bile salts with the hydrophobic end facing towards the globular of fat. The hydrophilic, water-loving, water-accepting, those hydrophilic ends are now facing outward. And so the hydrophilic ends are going to mesh with the hydrophilic ends of the phospholipid bilayer. And they can sneak that fat globular that has now been broken up and emulsified into these smaller pieces. It can kind of sneak it into the cell and have it be absorbed through or into the lacteal vessel of the lymphatic system. All right, and then again, it's going to go to the subclavian, specifically probably more than likely into the left subclavian, and enter back into the bloodstream. If you do not have enough bile being produced, your body's not going to absorb the fat. And if it doesn't absorb the fat, it's going to pass right into the feces, and you're going to have very different characteristics of your feces. And that's all I'm going to say about that. Your vitamins, water-soluble, your B complexes, your Cs, they're going to use diffusion to go right on in. And that's going to happen all along, especially within your minerals, all along the length of the small intestines. Water is going to be absorbed as well as all along there. Keep in mind, you start with about 9 liters of water as it enters into the small intestines. By the time, follow this, let me just put this up here real quick. You start with about 9 liters of water going in to the small intestines. you absorb about 8 liters along the length of the small intestines, so that by the time you get into the large intestines, you're down to about 0.8 liters. Of the 0.8 liters that enters into the large intestines, this is water, only about 0.2 liters actually get released through the feces. And so you've got a massive amount of water that is reabsorbed along the length of the small intestines. One final little push of absorption of water in the large intestines, and then the rest release. The body is a water conserving beast. That's what it's supposed to do. Conserve water. We are water-based. 55, 60% of us, of our bodies are water. And so conserve as much water as we can, and the body seems to maintain it, be a little bit more happier. Fat-soluble vitamins. If you are on such a lean diet that you have very little fat going in, and you are taking fat-soluble vitamins such as A, D, E, and K, you're wasting your money. Your body needs to have a little bit of fat in it, because the fat-soluble vitamins need the fat in order to be absorbed. So if you cut out fat completely, and you're taking vitamin A, vitamin D, and vitamin E, and vitamin K, give me your money, because you're wasting it giving it to the pharmaceutical companies. Those vitamins need fat in order to be reabsorbed into the system. Just a word about the large intestines, as I've already mentioned. Again, no real tricks here. You've got the cecum right there. Here is our ileocecal sphincter. Again, controlling the flow of chyme going in from the small intestines into the large intestines. And as I've already, I think, made mention, here's your ascending colon. Here is your transverse colon. Here is your descending colon. Here is your sigmoid colon, leading down into the rectum, passing through the anal sphincter, and passing out through the anal canal and the anus, taking all that feces, all the leftover, non-absorbed particles of what started out as a cheeseburger or a chicken sandwich or spare ribs or Western cheeseburger with extra large fries and a side of apple pie with a big old thing of Coke or Pepsi. It takes that and transforms it and reduces it and absorbs the water and the nutrients and the vitamins and the minerals and the carbs and the proteins and the fat, and what's left is your feces that's produced. Remember, bilinogen is the byproduct of the breakdown of bilirubin, and that's what's going to give feces that characteristic color and odor that we see. It takes about 24 hours for the digestive system to work all the way through the entire system from beginning to end, from mouth to anus. In a normal person, about 24 hours or so. In people that's not normal, it's going to take longer or might take shorter. Just a couple little words about some of the intestinal things that we run into. There's about 800 different bacteria or flora that's found throughout the intestinal tract. Ladies, I hate to tell you, but every person contains about 7 to 10 liters of gas within our intestinal system. We expel, we release into the environment about 500 milliliters a day of gas. Ladies, you can't deny it. Even you release about 500 milliliters of gas per day. Don't pretend. Again, it takes about 24 hours to prepare that feces, and you can kind of see the breakdown of what it is. About 75% of the feces is going to be water. 25% is going to be solid. Of that 25%, 30% is going to be bacteria, 30% undigested fiber, 15% fat, and the rest of it's going to be epithelial cells, it's going to be mucus, it's going to be protein, it's going to be things that are going to be not digested. 30% undigested fiber, right? Things that are cellulose in product, a lot of your roughage, anything high in fiber. So a lot of your vegetables are going to be in that category. A lot of your whole wheats are going to be in that category. Our body just can't get in there and break down that celluloid structure that's making up the cells. And so what it does do is we classify it as roughage. And so it helps to remove and keep the colon clean. It keeps the intestinal tract overall very clean. We're getting rid of bacteria. All right, they're going to get caught up in the mix. They're going to die off. We need to get rid of it. I can't stress the importance of this. Bacteria are our friends. All right, 800 different species of bacteria, and that's probably underestimating the number of bacteria different species of bacteria that we have in our stomachs. We are living in a society, and I don't mean to preach about this, but we are living in a society that are raising germaphobes. We are so hypersensitive to bacteria. Bacteria have this negative connotation within our society. They're bad. They're evil. We've got to kill them. 99.9% effectiveness, killing bacteria. We need to realize that without bacteria, these are beneficial species of bacteria, 800. I would say probably into the low thousands. Different species of bacteria are residing in you, and you need them in order to have proper digestive function. And yet, as a society, we strip them away. And we wonder why people have as many digestive issues as they have. Because the bacterial balance is not there. The flora is not there. The fauna is not there. We don't have the breadth of bacterial species that we need to have to function as humans. For God's sakes, if you look back to the beginning of evolutionary time, we more than likely started because of bacteria. Bacterial RNA getting into a DNA structure, and bam, you've got the ability to form proteins. We are based on bacteria. Let the bacteria live. It's okay to get a little dirty. Sorry for pontificating. But that is so important in order for us to understand proper intestinal health. We need those bacteria there. We really, really do. And we spend so much time and energy advertising through our pharmaceutical companies and alike that bacteria are bad. We need them. They are our friends. Let them live. And then sloughed off epithelial cells and extra mucus and protein and all that kind of good stuff is also part of that feces preparation. We have come to the end of A&P 2. If you have heard this WIMBA, barring any ability or time to go into reproductive systems, if you've heard this WIMBA, you have completed the information needed to be learned for A&P 2. Congratulations. You have survived. Possibly. Still have an exam to go. On that note, as always, if you have any questions, feel free to email me or write them down and ask me in class. I'm always here to help you. My greatest concern is that you learn the material, know the material, and be able to apply it. And hopefully that's what you have been able to do throughout this A&P 2 course. This is Professor Young signing off and saying happy studying and see you in the classroom.